All right, thank you everybody for joining us today for the seminar. I'm really excited and really proud to introduce our postdoctoral fellow in the T32 training program, Dr. Kim Darlington. She's been in the lab for close to over a year and a half now and has been doing some amazing work looking at how we can utilize multi-omic sequencing data and find a target or targets that can potentially be actionable in the context of understanding mechanisms behind disease and potentially even therapy. Dr. Darlington graduated from Duke University with a BS and completed MD, PhD there as well, as part of the MSTP program, and then joined UNC as part of our T32 basic science training program. But she's been very active clinically over the last year and a half. I've seen her in clinic. She's working actively with our clinical faculty, prolifically publishing, and at the same time, working in the lab and coordinating the efforts of many scientists, not only in the lab here, but also nationally. And today's work sort of will demonstrate the culmination of that effort. It's also a really amazing training vehicle. And I know there are some people in the audience that are translational, for them to understand how you can actually approach translational medicine and make a career out of it, and impact patient care as well. So with that, Kim, the floor is all yours.
Well, thanks for that very kind introduction, Shavad. I'm very grateful for the opportunity to share the work that I've been doing over the past year and a half in Dr. Steig's lab, and really describe some what we'll call, works in progress, but, culminating it's a really interesting story that we hope to come to a conclusion to in the next few months. So the title of my talk is Increased Cell Adhesion Molecule 1 or CADM1 Results in Aberrant Mucosal Immune Responses in Patients with Ulcerative Colitis. And I hope to sort of convince you of this story, describing a signaling axis that involves a key microRNA and the cell adhesion molecule in IBD patients, particularly in ulcerative colitis, but also in Crohn's disease patients as well. So just to get started, some very brief background, since I know this is familiar to a lot of this audience. But basically, just thinking about the pathogenesis of inflammatory bowel disease, we recognize it as being very akin to a lot of chronic illnesses that don't have sort of one sort of slam dunk target or, otherwise definable genetic susceptibility, for example. And that it's a constellation of the environment and genes and inflammatory responses that all interplay to sort of lead to the disease as we see it clinically. And particularly for inflammatory bowel diseases and ulcerative colitis, which I'll focus mostly on, as we recognize it as being an immune-mediated chronic condition and clinically defined by periods of remission and flare. And among sort of these four aspects, I'm particularly interested in sort of in the role of the impaired epithelial barrier function, which we know to be sort of pathologic in ulcerative colitis, and how that interplays with the immune response to additional injurious stimuli, which we also know to be abnormal in patients with IBD. And so the goal of my project that I'm going to talk about today, and sort of was defined when I came into the lab, was really to focus on ulcerative colitis patients. But again, I'll sort of allude to the fact that some of the findings that we've come upon are akin to or relevant to Crohn's disease as well. But basically with the idea that we wanted to understand the molecular phenotype of these patients and really focusing on a unique subset of ulcerative colitis patients, those that have basically failed all medical therapies, and what a classification of patients that we refer to as medically refractory, meaning that they've undergone several different medications, usually steroids, a variety of biologics, for example, and had to come to the decision that the only pathway forward to control their disease is to undergo a colectomy or surgical resection of their colon. And so this project is sort of focused on defining some of the molecular phenotype of that specific population, and I'll talk more about that in a second. And really the goal, the downstream goal and really the pipeline dream would be to identify potential novel therapeutic targets for this very at-need population, but really more practically also thinking about how we can use the drugs that we do already have available and the pathways that we know to be active or targeted by those therapies, and potentially use them in these contexts as well. So I'll start by just giving an overview of the cohort or the population of patients that are primarily used for this study, but again have been sort of supplemented by additional analyses and other data sets that we have as well. So this cohort involves a collection of patients. We have 46 that were identified that met the following criteria. Basically, they had to have a diagnosis of ulcerative colitis, and then needed to have medically refractory disease, meaning they had to go on to have surgery, a resection of their colon. And we had to have stored samples between the years of 2012 and 2019, and those years being the cutoff, with 2019 being important to allow for appropriate clinical follow-up after their surgery. And the other important factor that we considered was that they had to have impaired, inflamed, and non-inflamed surgical samples stored. And why this is important is because it allowed us to do a variety of comparisons that really allow for sort of robust controls. And our controls include within the same individual, sort of comparing their inflamed or active tissue with a non-inflamed tissue, and then also comparing the findings that we discover in the active tissue with non-IBD surgical samples. So patients that also had to have a surgical resection for another reason, particularly diverticulitis and colon cancer being the indication in those controls. And there's about 20 of them. And with these patient material, we've done a variety of analyses, which I'll go through throughout this talk. The first being some comprehensive transcriptomics. So looking at both small RNA sequencing and mRNA sequencing from preserved tissue. But then also using the FFPE tissue to look at some additional analyses, particularly focusing on spatial phenotyping of the tissue microenvironment. And we have started some spatial transcriptomics, but I won't talk much about that today. But really with the objective, as Shahzad was alluding to, of looking for sort of tissue that could be taken advantage of in sort of a setting even outside of the context of this study, where we can do analyses that are relevant to sort of clinically accessed tissue or clinically collected tissue that could then be used for directing further therapies, for example. So again, using this cohort, we decided first to take a look at the microRNA profile. And the reason that we started with the microRNA profile, because it has several unique features, which allow it to help us to understand sort of the molecular phenotype of these patients. And the first being that microRNAs, just in general, are non-protein coding transcribed RNAs, but because they're transcribed, allow us to do a global assessment of the expression, similar to the way that we do mRNA analysis. The other appealing part of microRNAs is that they have the ability to target multiple different genes, so have the potential to be master regulators that could potentially define a disease process or a disease subset. And then finally, they have this ability to negatively regulate gene expression through mRNA degradation or translational repression. And that activity in particular is the one that we were most interested in because of the ability to point to a potential mechanism, again, that could be disease defining. So again, we started by doing a profile of the microRNA of our patient cohort of the UC patient samples, both in their inflamed and non-inflamed tissue, and compared them within each other, but also to the non-IBD samples that we had. And so the first data that I'm showing you here is the microRNA profiling, and basically demonstrating based on the hierarchical clustering that the microRNAs that we identified to be differentially expressed in ulcerative colitis stratified them as compared to non-IBD. So basically this map is showing you that in pink we have the UC samples and in green the non-IBD samples. And again, these differentially expressed microRNAs nicely separated the UC samples from the non-IBD controls. So then we wanted to look deeper at these microRNAs that are differentially expressed, again, in the inflamed tissue as compared to the non-inflamed tissue, but also in compared to the non-IBD samples. And so on the left, I'm showing you a volcano plot of all of these microRNAs that were differentially expressed between the UC patient samples versus the non-IBD patient samples. And the one that I've highlighted here was of particular interest is microRNA 375. And empirically, part of the reason why this one was of particular interest is because it was one of the most robustly and significantly decreased microRNAs in our entire analysis that was differentially expressed between the UC samples as well as compared to the non-IBD samples. And that's what's indicated on the volcano plot here. What's also interesting on the right is that I'm showing the normalized counts of microRNA 375 across the different conditions. Non-IBD in light blue, UC non-inflamed tissue in green, and the UC inflamed tissue in pink. And what you can see is that even in the non-involved sort of healthier or uninflamed tissue, that there is a relative decrease in the UC non-inflamed tissue, but that decrease is even more profound in the UC inflamed tissue. So this suggested that microRNA 375 could be an important differentiator in our disease state as compared to the control states that I described. And what's also interesting about this finding, so I mentioned, we were looking in our UC cohort that a collaborator from Dr. Praveen Sathupathy's lab at Cornell has recently identified that a similar relationship exists in Crohn's disease patients. So this is from one of his grad students, Alex Shumway, who has recently submitted this paper basically looking at treatment-naive pediatric Crohn's disease patients, a pretty large cohort, and looking across the expression in the colon and the ileum of Crohn's disease patients as compared to non-IBD patients. And again, the non-IBD is in green and the Crohn's disease in light blue. And in both the ileum and the colon, you can see that the Crohn's disease patients have decreased microRNA 375 expression. So not only are we seeing this robust decrease in ulcerative colitis patients, but this is also true of treatment-naive Crohn's disease patients, suggesting that it could be a sort of a unifying feature in these IBD patients in pathology and the colonic inflammation, as well as the ileal inflammation that we see in Crohn's disease. And then finally, in the literature, I actually think this is quite exciting that it actually seems to be a reproducible target in others' hands and others' labs and other cohorts, that there has been sort of two key publications that I was able to identify describing microRNA, but in 375, but in pretty small cohorts. So on the left, describing findings in human subjects. So Wu as being the lead author in 2019, they described a small cohort of UC patients with an N of 14, and again, a small cohort of Crohn's disease patients with an N of 12, basically showing that miR-375 is decreased in those colon tissue of those patients as well. And the mechanism that they pointed to is that miR-375 potentially negatively regulates TLR4. Along these lines, and a little bit of an older paper, using the IL-10 knockout mouse model, which I think everyone here is pretty familiar with as being a model of colitis, basically showed that in IL-10 knockout colon tissue, miR-375 is decreased in those mice that have pathology consistent with severe disease as compared to those that have less severe disease. So a histology grade of 3 as compared to 0 or 1. So suggesting that, again, across different model systems, that this miR-375 decrease is reproducible and relevant. So taking these findings together, this sort of led us to the question, well, for one, what are the functional consequences of this miR-375 decrease? And we see it right now, we're just defining an association. What potentially is the role of miR-375 loss, and what sort of potential pathways and mechanisms is it regulating? And then why is this happening? What cells are responsible for this difference would be sort of the second key question that we're sort of focused on answering. So the first thing, just to give a general overview, wanted to get a picture of what's basically happening in our tissue sort of at the global RNA level. And basically what I'm showing here is pathway analysis of the transcriptomic differences in the UC inflamed tissue versus the non-inflamed tissue. And some of this is more just a reassurance or, sort of confirmation of what we'd expect, that when we are looking at the RNA profile in our inflamed tissue versus our non-inflamed tissue, the majority of the pathways that are active or upregulated include pathways that are involved in extracellular organization, which are highlighted in red, and several chemotaxis pathways, again, that are increased in the inflamed state. So again, not sort of unexpected, but at least to be consistent with the fact that these are the type of pathways that, at least at an RNA level, that we're seeing to be upregulated in ulcerative colitis disease tissue. So then this raised the question, well, what role potentially does miR-375 have in these processes? And basically, we wanted to test the hypothesis that genes predicted to be negatively regulated by miR-375, as defined as having a conserved miR-375 binding site and increased mRNA expression in UC inflamed tissue relative to non-IBD, non-inflamed tissue, these genes play a central role in the pro-inflammatory disease state. And so basically, to test this hypothesis, we were able to use this algorithm designed again by Dr. Sethupathiy's lab, which is called miR-Hub, which basically takes advantage of a series of algorithms looking at these conserved binding sites and comparing that to our global RNA profiling. And again, our goal was to identify genes that have this conserved miR-375 binding site, but then increased expression in the inflamed ulcerative colitis tissue. What was exciting is that we identified about 200 genes that were differentially expressed in the ulcerative colitis tissue, particularly in the inflamed tissue, again, that have these binding sites. And here I'm listing a few. Basically, I'll go on to describe the pathways in a moment, but several of these were particularly interesting, among them being genes that we know were involved in chemotaxis and inflammation, like the MMPs. Also PDPN, which we know to be increased in activated fibroblasts, which I think is particularly exciting because I think the role of activated fibroblasts is really emerging in IBD. Won't be the focus of my talk today, but I still think is significantly interesting. And then TGF-beta signaling as being highlighted here, as well as other chemotaxis molecules or signaling molecules like CXCL1. And then finally, this one cell adhesion molecule, CADM1, which I'll spend the rest of my talk sort of describing. But just to go on to say that we also did the pathway analysis amongst these genes. And so again, this is just looking at the miR-375 target genes in the inflamed tissue. So those, again, that had the conserved miR-375 binding site, and looking at the sort of categories or pathways that these genes fall into, that those genes that are regulated by miR-375 similarly are involved in extracellular structure organization, extracellular matrix organization, regulation of cell migration, and cell motility as well. So not only do we see these differences on a global scale in the involved UC tissue, but we also see that miR-375 is regulating similar pathways with these sort of gene categories being defined. All right. So now that I've shown you, just to sort of catch up where we are so far, I've shown you that miR-375 is decreased in the UC tissue, the inflamed tissue, particularly as compared to the non-inflamed tissue, and as well as compared to the non-IBD tissue. I've also highlighted that cellular and chemotaxis pathways are important, or seem to be more upregulated in the severely diseased tissue. And the number of genes are regulated by miR-375 that are involved in these pathways. And one of them in particular that we were particularly excited in, again, is this cell adhesion molecule, CADM1. And CADM1 is particularly interesting for a number of reasons, which I'll go into throughout the next slides. But first of all, that it sort of meets these criteria and that it's involved in these pathways, which we believe to be really important in defining the disease state. And again, it also has this criteria of being regulated by miR-375. So we've identified that CADM1 has a conserved miR-375 binding site in its 3' UTR, but it also has these other features that make it appealing as well. Basically, it is has been defined as being a member of the immunoglobulin superfamily. It does have a role, as previously defined, in mediating cell-cell adhesion in a calcium-dependent manner. What's also super interesting about it is that it can be shed from the cell surface via the activity of proteases. And taken together, this makes it a really attractive drug target, being one of the many reasons why I think it's interesting to study. And I'll go on to describe this a little bit in more detail, but it's been described to be explained or expressed in a variety of cell types, including neurons, epithelial cells, immune cells, that are sort of viewing it as a sort of open box to discover in the context of IBD. So to summarize where we've gotten so far, just wanted to sort of formulate my hypothesis going into the next experiments. So basically, our hypothesis is that decreased miR-375 expression resulted in increased inflammatory state in ulcerative colitis, and really in Crohn's disease as well, via increased CADM1 expression. And by function of miR-375, this really means that when there's a lack of miR-375, there is less mRNA degradation, which leads to the increased mRNA detection or expression that we can observe in the disease state. So we wanted to test that hypothesis by looking at the mRNA expression of CADM1 in our different states. And on the left, I'm sort of reminding you of the data that I've already shown you, looking at the miR-375 levels in the colon tissue. So again, the non-IBD is in light blue on the left side, showing the highest level of expression, and the inflamed UC tissues in light pink, showing the lowest level of miR-375 expression. And on the right, I'm showing the CADM1 mRNA in the colon tissue. So the same tissue that we used for the microRNA analysis, now looking at the mRNA analysis. Now you can see that there is a robust increase of CADM1 mRNA expression in the inflamed tissue as compared to the non-IBD tissue. So we then can take this analysis one step further and look at sort of the relationship across individuals between the miR-375 expression and the CADM1 mRNA expression. So on the bottom, I'm showing the log-transformed miR-375 expression on this graph on the left, and CADM1 expression, which is also log-transformed on the y-axis, and basically showing that there is a negative correlation between CADM1 expression and miR-375. So again, supporting this hypothesis that when there is a lack of miR-375, there is an increase of CADM1 mRNA expression. So then this takes us to the next step. So now that we've shown that there is this relationship between miR-375 and CADM1 in our experimental model, we still wanted to ask the question, again, what are the sort of the downstream effects of this, and what cell types could be important in mediating this process? And one fundamental piece of starting to investigate this question came from a exciting collaboration with the Poi Lab, which is out of Hawkins, which has studied this relationship between miR-375 and CADM1 in a model of type 1 diabetes. And in their mouse model, they had basically defined several different targets that are regulated by miR-375, but had been really excited about CADM1 for a number of reasons, which I'll go on to show you in a second. But all to say that, you know, even in our data, amongst all of these targets, CADM1 really being the most appealing target. But again, I also wanted to highlight here that really just demonstrating the signaling axis as being relevant to a number of inflammatory disease conditions, again, type 1 diabetes being the one that the Poi Lab studies. So this is additional data from the Poi Lab. So sort of going along this thinking of trying to figure out, well, we've seen this relationship between miR-375 and CADM1, what is sort of the downstream effect, or what cells could be involved? And Dr. Poi's lab has been really excited about the role of CADM1, particularly in macrophages, in his model of type 1 diabetes. And so here I'm showing you a single cell sequencing from the pancreas of non-mice that develop type 1 diabetes. And basically what they're showing is that in the expression that's highlighted in red across these UMAPs, that there is the specific increase of CADM1 in a population of macrophages. And these data have led to the hypothesis in their model that CADM1 is expressed in macrophages as well as on their alpha beta cells. And that expression leads to recruitment or increased interaction with CD8 positive T cells. So now they've sort of focused our thinking about the role of innate immune cells, particularly macrophages, how they could interact with epithelial cells and immune cells as being important mediators of this sort of signaling axis that I've been describing, that we've been discovering in inflammatory bowel disease. So now that I've shown you some single cell data, apart from doing our own single cell experiment, there has been some really exciting data just published this month in Immunity by a group from Boston, basically describing the largest cohort of single cell data, looking at various components of the colonic and ileum samples. So basically, I'll describe this in a little bit more detail. So this paper was published, again, just this month, and they looked at single cell sequencing from over 70 patients. This included healthy patients as well as patients with Crohn's disease. And they looked at, so basically the cell composition from areas that were, again, defined as healthy, but also areas within the disease, the Crohn's disease patients that were non-inflamed and inflamed, and then subdivided their different single cell populations across epithelial components, immune components, and stromal components. And really, this is an impressive sort of mammoth effort, sort of characterizing these different cell types. And I think it's really created a nice resource for the IBD community to understand more about the different cell types that could be mediating this disease. So basically took advantage of this very recent data to take a look at some single cell data using an application through the Broad Institute, which I'm sure several of you are familiar with, just basically looking at different gene expression across their different cellular components. And so what I'm showing you here is the profiles of CADM1 expression across different cellular components. So the first column is showing you the epithelial cell component with the CADM1 expression on the top and the gradient from gray to red, and below it, the cell map showing the different epithelial cells. In the center is the immune cell component, again, the graded red, showing the red showing the highest intensity of the CADM1 expression, and then below that, the UMAP of the different immune cells. And then finally, the column on the right, again, graded red, showing all of the CADM1 expression across the different stromal cell components, and the MUMAP, again, defining the different stromal cell subsets. And so, on first view, you can see that CADM1 is pretty diffusely expressed. Particularly, we do see some areas where it could be expressed in epithelial cells, but there does seem to be sort of more enrichment or a more intense, focused, cell-specific type expression, we believe, in immune cells and stromal cell components. And based on what I've described already, we're really excited about the potential immune cell components, though we recognize that CADM1 is expressed in a variety of other cell types. So just to take a step further, I wanted to look specifically at the innate immune cells, again, because we have seen in other models that particularly macrophage-specific expression of CADM1 seems to be particularly important. So here I'm showing you in the abbreviated UMAPs. So on the left, again, is from gray to red, is showing the intensity of CADM1 expression. And on the right, I'm showing you the UMAP with a subset of the innate immune cells. And basically, you can see that there is a range of expression of CADM1 across these different cell types, with both macrophages and dendritic cells expressing some CADM1, and that particularly this population of DC1s, which is shown by this dark green, having a very intense CADM1 expression. But again, sort of this collection of macrophages here in the middle, also expressing CADM1. So again, just to emphasize that a variety of cell types express CADM1, and innate immune cells are one of those cell types and can be an exciting avenue of interest for further study. So far I've shown you some data from our collaborators lab, again, that looks at type 1 diabetes and found this association with CADM1 and macrophages. We've now looked at single cell data from the Broad Institute, looking at CADM1, being expressed in a variety of different cell types based on single cell data, and very, very new data that was just published. So now I wanted to take a look sort of to understand how this could relate to our model, and particularly our severe phenotype of ulcerative colitis. And as a first step, we basically wanted to understand the cell composition that defines sort of the severe ulcerative colitis phenotype that we're seeing, since, among the different cohorts of ulcerative colitis patients or even IBD patients, it's unique that we're looking at a surgical subset of patients. And to do that, our first step was to do some spatial phenotyping, particularly looking for different cell composition and some downstream analysis looking at cell-cell interaction. And again, we used this approach. It was basically defined as spatial phenotyping with the purpose to delineate, again, the cellular composition really in intact tissue, so FFPE tissue, and spatial organization that could suggest these differential cell-cell interactions. And so we used FFPE tissue from a subset of our UC surgical samples and stained it with 24 cell markers to define these different subcellular components. And our analysis looked both at cellular composition and the relationship to nearest cell types. So before I get into the data that we sort of produced from this experiment or this analysis, I wanted to introduce you to what this data is going to look like. So basically, the primary comparison that we're doing in this experiment is to compare the relationship between epithelial cells, sort of knowing that the epithelial cells are a key driver of the epithelial barrier or key components of the pathogenesis of IBD, and the relationship to the other inflamed cells that are within the tissue that we're examining. And the data is going to look like this schematic on the left, where the non-inflamed tissue is represented by red and the inflamed tissue is represented by purple. And in these violin plots, sort of the longer the distance, the higher these more extended distance is a greater distance to epithelial cells. And so the shorter the distance, sort of inferred then reciprocally, is the closer relationship to epithelial cells. So basically, on our survey of our tissues, basically we're able to identify that there were several populations that had a statistically different relationship between epithelial cells and the inflamed tissue as compared to the non-inflamed tissue. So the ones that are statistically different, sort of highlighted in red, but also that we have the CD4 regulatory T cells. But there are a few populations that stuck out to us. The first being M1 macrophages, showing a decreased distance between the epithelial cells and the M1 macrophages in inflamed tissue as compared to non-inflamed tissue. And similarly, there's a shorter distance between dendritic cells and the epithelial cells in the inflamed tissue as compared to the non-inflamed tissue. So again, while these cells aren't necessarily the closest ones that are interacting with the epithelial cells, they are statistically different or differentially distance from the epithelial cells, suggesting that they may be playing a pathogenic role in the inflamed state as compared to the non-inflamed state. So then we wanted to ask sort of downstream functional assessment of these relationships. And the first experiment that we wanted to do was an in vitro experiment, really starting with looking at macrophages, sort of again, knowing that innate immune cells are involved in this process and sort of established by the POI lab as being macrophages as being important mediators. Again, being able to ask these questions in vitro, really, for one, testing the hypothesis now that decreased miR-375 results in a pro-inflammatory state as mediated by the macrophages potentially. So for this experiment, we're comparing wild-type macrophages versus miR-375 knockout macrophages. And these macrophages have been isolated from the lamina propria of these respective mice. And they've been stimulated with heat and activated E. coli. And then the readout is the inflammatory cytokine expression. And you can see here that when there is decreased miR-375, as represented by the knockout, that there is increased expression of these pro-inflammatory cytokines. On the left, showing IL-1 beta, and on the right, showing TNF alpha. So again, the wild type is in green, the miR-375 knockout is in red, and you can see that there is an increase when miR-375 is absent in these lamina propria macrophages as compared to the wild type state. So this experiment was the lamina propria macrophages. We also wanted to take a look at the bone marrow derived macrophages, primarily because they're more abundant. Also, we can look at different conditions, and also to see if there's additional factors that could be assessed by looking at this condition. So basically, again, we took the wild type and the miR-375 knockout bone marrow derived macrophages, and now in this case, stimulating them with LPS. And similar to the lamina propria macrophages, we can see that when we stimulate the miR-375 knockout macrophages, that we can see an increase in these pro-inflammatory cytokines, including IL-1 beta and TNF alpha. And we also can see an increase in CADM1 expression, which is, again, sort of consistent with the hypothesis that the decreased miR-375 or the absence of 375 results in an increase in these pro-inflammatory cytokines and this our target of interest, CADM1. So now we wanted to then extend these observations that we've seen in vitro to in vivo models. And the first step was to look first, looking at taking advantage really of a specific mouse model that our collaborators have, where we are lacking CADM1 in all cell types that lack lyse-M, which include macrophages, but also other innate immune cells. To test the hypothesis that lack of CADM1 expression in macrophages or other innate immune cells would be protective against a model of DSS-induced colitis. And DSS being the chemical colitogen that I'm sure many of you are familiar with as a mechanism to induce inflammation. And basically looking at a 7-day time point, again, with the thought being that when CADM1 is absent, that there will be a decrease in inflammation and potentially a decrease in the severity of this model. So at this point, these are very preliminary data, but at least suggestive. It's purely looking at change in body weight, that when you look across these mice, that those mice that lack CADM1 are protected against DSS in a measure of change in body weight. And again, these are very preliminary data, and we're actively repeating this experiment, but at least to say, when you compare the wild type DSS and the CADM1 knockout DSS treatment, that there is a general decrease in the change in body weight in the CADM1 knockout, suggesting again that they are protected against this DSS-induced colitis model. And we are excited because we are actively sort of repeating this experiment with a larger cohort and intend to include sort of all the typical measures of the DSS model, including stool quality, change in body weight, not only as demonstrated here, but relative proportions, in addition to disease activity based on histology. So that is rapidly forthcoming, and we're really excited about that data. But at least to say, in this initial experiment, was supportive of the hypothesis and suggestive of this relationship that we were excited about. So taken together, I hope I've convinced you of these two aspects of the model that our work has sort of come to support. The first being sort of the very sort of molecular level, demonstrating that miR-375, decreased expression of miR-375 results in increased CADM1 mRNA expression. And that we've seen this particularly in ulcerative colitis patients, but really the same relationship exists in Crohn's disease patients as well. And then at an intracellular level or intercellular level, that increased CADM1 expression, we believe, mediates a pro-inflammatory state. And we're still in the process of investigating exactly how this happens. Again, we know that CADM1 is expressed in a variety of cell types. Epithelial cells, immune cells, and stromal cells is all being part of that process. But again, we're really excited about the potential role of the immune cell component as being part of this paradigm. So I wanted to shift gears for just a minute and talk about some of the ongoing work and future directions which we're taking this work, the current state. So the first, so I noticed that Dr. Chokonagi is on this call, and I'm sure Dr. Sartor is as well, but really excited about a new avenue, and thanks to them for enabling us to sort of test this hypothesis or test this experiment of targeting CADM1 in our IL-10 knockout mouse. And so really the goal of this experiment is to not only define the pathophysiology of CADM1, but even potentially highlight a novel therapeutic target. Basically using mice, the IL-10 knockout mice, and I'll describe the experiment in more detail in just a second, using an anti-CADM1 antibody and comparing it to control antibody, and then determining disease activity at the end of this experiment. So basically, the hypothesis that we're testing now ongoing for the past week is that inhibition of CADM1 will result in decreased microbe-induced inflammation in the IL-10 knockout mice. So basically, we've taken these IL-10 knockout mice and given them a CADM1 antibody and colonized them with human-derived fecal slurry with an FMT approach. And now we're going to continue to treat them with the CADM1 antibody and then assess them for disease activity at the end of the experiment. Again, sort of testing the hypothesis that inhibiting CADM1 could potentially decrease the inflammation that we observe in this model. And the other thing that I'll highlight is that this, again, is very new data that we're trying to understand sort of what cell types are expressing CADM1 in the IL-10 model. I've shown you some data that miR375 is decreased, but where potentially is CADM1 being expressed. And so this is very much just like hot off the presses data, but it looks as though at least that some of the subepithelial cells, whether they're macrophages or other innate immune cells, are responsible for expressing CADM1 based on some immunofluorescence that we've recently done. So on the left, I'm showing you the DAPI and CADM1 signal, and on the right, showing you the specific CADM1 expression. And it does seem to be that there is basically subepithelial cell expression of CADM1, sort of again highlighting that potentially either the stromal or innate immune cell component could be really the driver of the difference in expression and inflammation and that we see in these models. So in summary, throughout this talk, I've shown you that miR375 is decreased in ulcerative colitis and in Crohn's disease patients as compared to non-IBD controls. We've also observed that miR375 has several potential targets, one of those being CADM1, which we've seen in type 1 diabetes, and now the first to see it in IBD. We've shown that CADM1 expression is increased in ulcerative colitis patients as compared to controls. I've also shown you that miR375 expression in LP macrophages may result in altered immune response, as demonstrated by the increased cytokine expression, and that really CADM1 may play a role in innate mucosal immune responses as well, demonstrated by the other in vitro experiments and the lysine-free preliminary data that we have so far. So this has led to some really exciting future directions and some ongoing work that we're completing over the next few months, and it's already been started. You know, one of the questions, of course, being what is the cell type expression of the microRNA, miR375, and its genes of interest, particularly CADM1. And I've shown you some very preliminary data looking at the CADM1 immunohistochemistry. We're also doing in situ hybridization looking for miR375 expression. We have some preliminary data on spatial transcriptomics for cell-specific transcript expression. Particularly, we've seen that CADM1 does seem to be increased, again, sort of in that assay in the severe inflamed tissue. And now we're working to define what other transcripts are also expressed with it. And then finally, as I mentioned, the immunohistochemistry for CADM1 and associated cell surface markers. So I showed you the very preliminary data of CADM1 in the mouse tissue. We're doing similar experiments in human colon tissue to define the expression of CADM1 there, and then also multiplexing it with other cell surface markers to further define which cell types could be expressing CADM1 in our patient samples. And the final really exciting ongoing work is the functional validation of these gene targets, really using our in vivo models. And again, the ongoing experiment with the CADM1 lysine-CRE DSS model, which the POI lab has been helping or primarily leading that. And then locally doing the IL-10 knockout experiment using the anti-CADM1 antibody to determine the role of targeting CADM1 in this model of colitis. So with that, I want to, I owe a lot of acknowledgements to many people that have been involved in sort of this mammoth collaborative effort. Of course, wanted to thank Dr. Sheikh for his mentorship in this project and allowing me to do a truly translational project, which has been very exciting. And all of our lab members, including Wenbo Lee, who I showed some of his in vitro data. The Pathogenesis lab has been helpful for the microRNA analysis and shared their miR375 knockout mice. The POI lab has been instrumental in sort of extending their observations from type 1 diabetes to IBD. And of course, the Fury lab for their mentorship and doing all of the transcriptomic analysis. And finally, wanted to say thank you to the Starter Labs and Dr. Chuck Conaghy for helping facilitate the IL-10 knockout experiment, as we think that's a really exciting avenue and should have results from that coming here shortly. And so with that, I will take any questions that you have at this time.
Kendall, if you want to unmute yourself and ask your question.
Kim, thank you. That was great. Fantastic. An avalanche of data coming our way in the next few months, and you really synthesized it well. Go ahead, Kendall.
Yeah, great talk. Really interesting stuff. One of the things that I thought would be fun to know more about is just kind of the molecular mechanism by which CADM1 might function in cell-cell adhesion. So is this, a lot of cell adhesion molecules form these homotypic interactions in trans to kind of facilitate the adhesion between cell pairs, but other molecules in the immunoglobulin kind of superfamily are also known to form heterotypic interactions and bind other kinds of cell adhesion molecules to kind of specify unique interactions between different cell types, for example. So I don't know if you can talk a little bit more about that and what's known about the binding capabilities of CADM1.
Yeah, that's a great question. I think you're thinking about this in a very unique way, but something that I've sort of explored, both talking with the POI lab and reading some literature about CADM1. But I do think that it, at least from their model of thinking about sort of the macrophage beta cell interaction, that does seem to be sort of a CADM1 interaction. But at least when it relates to macrophages and T cells, it's a CADM1 and a CR-TAM receptor interaction that might be relevant. Whether that means that CADM1 is then, forming dimers with other cell surface receptors to facilitate this, I don't know, but it's a super interesting question for sure.
This is Belfer. I echo Shahzad's point that this was absolutely beautifully described. Great data, but great presentation as well. So Kendall beat me to the question about the heterotypic interactions. I'm really excited to see your tissue staining. I assume that was that in situ hybridization there, or it was
that
in
was fluorescence immunohistochemistry there.
So it would be really nice to double label cells to see which cells it's emanating from. But kind of as a corollary of Kendall's question, we think of adhesion molecules a lot in trafficking of cells as well. And is there any evidence that there's endothelial binding that might target different organs or inflammatory conditions for circulating cells, in addition to your orientation on in the tissue itself?
Yeah,
that's it's almost counterintuitive, because if you had binding, if you had binding, it's not counterintuitive. I apologize. If you had upregulation in inflamed endothelium, it might also be a way of bringing cells into that, as in the alpha 4 beta 7 T cell trafficking.
Yeah, so I think at least from the single cell data that we have from that group at Harvard, there doesn't seem to be any definition of endothelial cell expression. So looking at normal tissue, there are, I think, some small signals for CADM1 mRNA in endothelial cells. I think it definitely will be relevant to pay attention to that when we're doing sort of our immunohistochemistry to see if we can at least see the protein expression of CADM1. As far as the in situ hybridization, we're focusing more on the miR-375, but the mRNA would be interesting as well. But your point is well taken that, trying to figure out how the cells are utilizing this cell surface transmembrane protein to sort of mediate these interactions, and with the sort of trafficking along the endothelial cells being one mechanism, certainly is interesting.
And Balfour, I just want to add that the Kim is actually co-stating for a bunch of markers for macrophages, T cells.
Right.
Just to this was just right. I think we just got this data yesterday, Kim. And
yeah,
then,
so we're just doing the single
yeah.
stains right now, but now we're moving on to the human tissue and then multiplexing all of them once we've decided on the right conditions, but definitely an ongoing effort.
And the one thing I would add is that in the NOD model, when they gave mice CADM1 antibody before they developed diabetes, that was protective. And so I'm wondering, and they didn't, I don't think they saw a difference in the, you decrease the inflammation in that context, whether they saw changes in numbers in CD8 T cells that are infiltrating the tissue. But they just found a decrease in islet cell destruction.
Right. They did show that there's a difference in interaction with CD8 T cells when when there is no CADM1 or the inactivating antibody is present. But that doesn't answer necessarily the question about the endothelial cells. And in their publication, they focus primarily on the islets. So it's not
right.
defined.
And it's what I'm saying is that the chance that it's preventing trafficking versus the direct inhibition at the site of action is probably the latter, is probably the mechanism.
Right.
Yeah. Well, cool stuff. I mean, it was really a tremendous body of tremendous amount of data here.
Absolutely.
And
I'm just
sure you're showing the tip of the iceberg.
Right. They're this is just one molecule in the context of the cool stuff we're finding. So if there are people that are listening and want to do a postdoc, come on down into the lab.
Yeah, I mean, I would say that the other targets of the MIR315 are also ADAM, for example, ADAM1, for example, and some of them could be balancing, and that some of those are more inflammatory, anti-inflammatory rather than inflammatory.
Right. And,
yeah,
and similarly, are really excited about potentially the role of the inflammatory fibroblasts and the way that MIR375 might be acting. Some of that sort of spatial data that I didn't get a chance to show is highlighting the role of activated fibroblasts. And I think we're just sort of, as you're saying, the tip of the iceberg, just getting to the tip of the iceberg of the activity that plays both in ulcerative colitis and in Crohn's disease.
Yeah. Well,
Kim, this is Sue. I have a quick question. Are you done, Balfour?
Yeah, go ahead.
Yeah, I echo fantastic talk. Maybe the diabetes publication would answer my question, but I would have a concern when you use anti-CADM1 treatment in vivo to be able to evaluate its effectiveness. This is always a question that we ask when we see these kind of papers. So how did they do that? And is it possible to cross IL-10 knockout mice with the Lysem-Cree conditional knockout that you've used and used those? It might be convoluted, but
yeah,
might be better.
Yeah, that is a great experiment that I think could be potentially complicated. So their readout, as far as I understand, because they haven't yet published these data, particularly with the antibody, they're looking for infiltration of macrophages and T cells within their pancreatic islets when they're treating with CADM1. So, it's a correlation at best. So we're using an IP injection, systemic injection, and trying to see if there is invasion of these markers. And then this opens the wonderful box of scientific inquiry to understand why exactly that's happening. And, I think you have to do that initial experiment to see if that effect exists. And then you kind of go down the path of trying to figure out exactly why. But that's sort of what the stage that we're at as well.
And then the question that everybody always asks, are these changes proceeding on board and a risk factor, or do they follow the development of inflammation?
Yeah, and I think, especially when we're looking, I kind of highlighted sort of the unique aspect of our cohort as being this really severe medically refractory phenotype. But I think, Balfour has highlighted this in the past as well. It's just, we don't know what's cause and effect at this point because we're really assaying the sort of end stages of disease. It's possible and likely that both are at play, but I think that's why we're doing the experiment, to see at least how it could be involved in promoting the initiation of inflammation as opposed to just the healing at this point.
Sue, those are excellent. Those amazing questions. We're definitely like the fall RO1 is going to be proposing IL-10 cross to the CADM1 lysempri. The second thing is, remember, we found the same association in treatment-naive pediatric patient samples as well. And so I think there might be, obviously there's a role in UC, it might be completely different in the context of Crohn's. So we have it across 3 different disease states.
Wow, yeah, great, fabulous.
Thank you.
You too.
I guess there are no more questions. We're at almost at 5:30. Kim, fantastic job. Thank you, everybody, for coming.
Thanks for all of your
talk.
questions and your attention. I appreciate it.
Absolutely. Great work, Kim. Have a great night, guys.